Thank you very much. Good afternoon. And it is a great source of joy to be here with this distinguished audience. For many years, we started becoming concerned with soil biology. And at Aapresid and focusing on sustainable agriculture, we started studying soil biology, not just physically and chemically, but also biologically, with a focus on carbon. It is quite pleasant, actually, to be here with speakers from Brazil and from Argentina who will show us some data. If you remember, last year we had Oliveira and Paraná showing carbon information, and we will see how that has evolved. And INTA has us used to this type of information. The case of João Carlos, we haven't had them here for many years, so it is great to have them here to tell us about the experiences in Brazil. And Mary Bartz. And it is not very nice to say the father of or the mother of, but Mary Bartz has a name that needs no introduction. If you want to Google Herbert Bartz, was one of the pioneers of no-till farming. I was born in 1972, and it was that year where he created his first parcels with no-till farming. So Mary will tell us more about their work on soil biology. So without further ado, let's hear from the experts. And we will hear Juca, João Carlos, in a presentation on carbon in Brazil. Thank you. Good afternoon, everyone. For me, it is an honor to be here, participating in this Congress. It is the first time I participated was in 1992, and I was received very warmly. So I'm very grateful to the organizing committee at Aapresid. The issue that we will be discussing today is the restoration of soil organic carbon stocks in Brazilian biomes. This is a result of a project that was initially sponsored by a European organization called Euroclima Plus, and a proposal of the Ministry of Agriculture which focused on different aspects, and the Brazilian Federation, that the project including 3 modules. So I will describe some of them, and then Marie will describe the other part. And so we asked a question in 2020. The situation that we had originally starting in 1972, we had our first hectares, you all know the story, and all the way to 2020, to the last census that was published by our colleagues at the Federation, with 7 million hectares that were involved in livestock and also sugarcane. But the issue was why in this whole area we have a barely 10% of no-till. And the 3 principles that are included, which are the total absence of tillage, the permanent cover, and the diversification and rotation of crops. So our challenge in this project was to show that we have the conditions to move forward and to have a reference in the sense that agriculture should be completely based on the no-till system. And I will try.
Vamos ser 3 tópicos.
So there are three topics: the methodology that I will simplify, and I will show the results. We have shown that the biomes are where the red dots are. We have 63 sites from latitude 7 to latitude 29. And on each site, we sought to show three types of uses of land. The native vegetation with pristine soil, undisturbed, the plow-based tillage soil that has been tilled frequently with advanced degradation, and finally, the no-till system soil. With this, we created and showed the contrasts and the intermediate points to determine where we are. The distribution of the points in the Cerrado and the Mata Atlántica, in the Atlantic forest. So the native vegetation has to represent the situation of each site. We have a plow-based tillage system that depends on the region, of course. For example, in the equatorial tropical region, it is different from the subtropical area. And this has an impact on everything that we knew, and also has a new equilibrium state called a dynamic equilibrium at a lower level. And with no-till systems, that involves three principles, like I said before. And this develops a new dynamic balance in the soil carbon. An example of how we took the samples can be seen here. NV means active vegetation, PBT means plowed-based tillage, and NTS is a no-till system. And it shows with scientific rigors and with the necessary replications how deep there is between 10 and 100. So what we reached was 3,402 of samples. We conducted several analyses, and you will have like an advance and a preview of this. We worked with enzymes, and we worked with undeformed samples with the capacity of water storage. Let's go to what interests us, which are the results. But before this, we conducted a large study that involves the different aspects that Dr. Lal made available to work on this at Ohio State University. So we created these areas where they have a group of similar components that happen together for us to be able to compare this with things that are at the same level, to be able to compare them. And with this, we involved environmental covariables, which are the characteristics of temperature, rains, sunlight, altitude, and this with 1 million data sets, where we created the different clusters. And what we did was apply this with a person that works with us called, the program was called Spectrum Clusters. And it's an algorithm that separates by latitude and making the climate variables interact. With this was created 13 climate areas. And it's very interesting because I will now explain what this means. And these zones are marked with those dots and those circles or with the ovals. And with this, we have been working in the following situation. The clusters, which are edaphoclimatic areas, define the basic groups based on the evaluation. And what we did was typify. Cluster 1 to 3 was the tropical equatorial climate in latitude 7. Then central, tropical, subtropical humid, all the way to subtropical humid and temperate. In order to do this, we used all the variables that we had available, such as mapping of climate areas by biome, by altitude, by temperature, by type of soil, and other factors. And based on that, we defined the limits and the characteristics. For example, for the equatorial tropical group, what were the differences? The differences were typification of the climate, altitude, and the characteristics of the winter. If they had a very dry winter with a long period of drought. With this, we were able to classify altitude 7, 8 to 13, and then all the way to 23. With this, we were able to characterize all of the soil climate zones or edaphoclimate zones. We used another program here called an integrator that groups together a lot of data and detects and identifies the areas with extreme weather and then establishes a median or an average. And we use that in order to develop the entire understanding because we had 63 sites. So how were we going to count things? How were we going to quantify it? These are the first results. Take a look. This is how we developed the accumulation of carbon based on latitude from 7 to 28 of latitude, and the 13 zones, equatorial, central, humid, temperate, and divided into tropical and subtropical. And we observed various major trends where there was native vegetation and all of the integrated variations. And so we observed two major trends. One, sandy soils with less carbon than acid soils. So where they had more carbon, we had first, where the latitude was higher, we saw all of the aspects that were associated with these climate variables. So they had a maintenance of the organic matter. All of these presuppositions were confirmed. And all of the areas in this second case, we separated it in clusters, and the trend was the same. Take a look. Where it increased, where it went down, and take a look at the red lines that you see there. The red lines mean an average and the upper level. With the tillage, the upper line, I apologize, but the microphone is having microcuts, and the same trend was observed.
When we look at the no-till system, we are starting to understand that some of these clusters, such as cluster 1, has 4 sites. So it's showing that cluster 1, limey, sandy oils, tropical climate, the mean temperature is 26 6 degrees Celsius. It's an oven. It's accumulating more carbon than in the native matter. This generated the first big conclusion. First, historical losses. What are historical losses? The carbon of the wild area minor the no-till. So in the 63 sites, we lost a variation from 34.5 to 60.4% of the total carbon. That's why we understand that no-till should not derive to these results. The historical gains based in 13 clusters was 48 to 103% of recovered profits. When we compare the wild area with no-till, we are close to 64.6 to 110%. We have to see that everything is a process of the average, that it's an analysis. It's an analysis that is not just focusing on the weighted average, but it's a direct comparison per group. This green ellipsis is very interesting, whereby all the properties have sites have a carbon level that is above natural vegetation. We apologize from the English booth, but the speaker does not speak Spanish clearly. He's speaking a mixture of Spanish and Portuguese. No-till in cluster 1, if you look at if you compare the chart, you will see that is just like in the case of native vegetation. So this provided this did not give us a very certain result as to where we are. These are the historical losses of the 63 sites. So the line, the mean line, the lower mean line is 30.4, and the upper line, dotted line, is 60.4 approximately. So you have two extremes, because we are not counting the extreme points. Is the response from a set of data. Why carbon and nitrogen are considered together? 3,280. There's a linear relation between these. So we are speaking about carbon. We are speaking. This is a different discussion. We're going to leave it for another day. And to me, this was the key point. Why no-till can accumulate more carbon? The beta-glucosides has a direct connection with carbon on the soil. So in the first graph, to the left, the soil is in the forest, and the next is under no-till, and the last is no-till soil. If you compare the activity of beta-glucosity is higher in no-till than in the forest, in the natural, in the wild. Because we are because there's carbon that stimulates the activity of the soil microbiota, and if we are tilling, if we are keeping the soil covered, if we are working on with diversification, we are storing carbon, and microbiota works more. It has more food. If it has more food, there is a direct relation with natural vegetation in the forest and in the area with tilling and in the area with no-till. This is the historical carbon restored. This is very interesting, showing 3 lines from 60 to 100% carbon restored. So what we are looking is that in most areas, chosen areas for no-till, are doing their job well. They are restoring, they're recovering carbon. What we used to think 30 years ago is now a reality. Look at this. This is very interesting. We did a scale. Out of 19 sites with over 100%, 16 sites between 80 and 100, 22 with 60 to 80%, and in 14 sites between 70 and 80. With this, we show that a no-till system based on its principles, and now I would like to repose not a new concept, but a new idea. If no tillage based on the 3 pillars, carbon, and also based on 3 pillars, quantity of contribution, quality of the contribution, and frequency of the contribution. If we work these variables together within us, it will be a win-win system. So this is the secret. No-till system associated to carbon harvest based on quantity, quality, and frequency. So with this, let me show you an example. We recovered carbon in cluster 1, that is tropical, equatorial, of 80 centimeters, sandy soils. Why? Because we have coverage, a dense coverage, with robust roots, aggressive roots, and deep roots that keep the soil covered. Have a look at the effect of the roots. They go well below 1 meter. The other situation, cluster C2, is a sandy soil. The transition 30-20% is the same. The system with grasslands associated to some pulses, but setting the roots up to 4 meters depth. And now there's this a comparison. 2 farms of the same producer. This is a farm of 44 years of no-till system based on corn, involving soybean, maize, dark oat, vicia, different crops. So if you look at this from 0 to 100, the green one, that is no tillage, is above the wild forest, recovering by 0.20, 0.40, and the total is 107.9%, compared to the other farm that has dairy livestock, and with this they get organic matter for silage, and that brings about a difference. You compensate with manure, however, you don't have the same effect of the dry matter. But a longer time with no tillage recovered, 0.20, 0.40, and almost 0.100. So these are things to discuss in the future. Why does it store more carbon in the case of no tillage? Natural pH on the soil, 4.5 pH in our case. Clay, aluminum with low pH, and the stabilization of organic matter is associated to acidity. So we started to understand first, roots of the different types make the difference. 60% of the accumulated carbon on the soil comes from the roots. Secondly, the green sources between the dry season has to be, needs the soil to be covered permanently. So here we are comparing farm in the Mato Grosso to one in the Amazonia. So with this we have roots, soybean of plus brachiaria plus corn. And another point is that clay oxide has positive charge, so carbon gets involved around. Then the management with common strategy, calcium remains as the main element. Here you have calcium with a higher quantity because these are clay type of soil, and the linear relation between calcium and carbon showing the interaction, the close interaction between these two elements. And Verde will control temperature, the physical barrier, and will provide carbon in depth. 60% of accumulated carbon from the roots. So the green bridge to cover during the drought season, in the case of clusters, 1, 2, 3, up to 7. So we have soybean, corn plus brachiaria, and stubble of brachiaria and soybean. So where are we? This is something interesting. There's a lot to be done. And here in cluster 1 to 3, we observe the variation, the amount of clay was 70% sand also. The stock in the wild forest, no-till and with tillage. And we did the calculation for historical losses, historical profits, and the capacity of recovery of 102.5%, and the restoration with reference to the wild forest, and the annual contribution of biomass, of carbon biomass, which means 20 tons of dry matter per year. And last, we calculated the sequestration rate and the conversion rate. So we are doing this for all the different areas. And in Article 2, we see the provisions, what the strategies we can develop in order to have a production system that is more stable, how we can mitigate risk, and where are we recovering more carbon? What do I have to intensify? And with this, next to the Amazonia, the stock is almost twice as much in the case of the forest, but the trend is similar. They recover 95.2%, 7.1 tons per year of carbon. Rates are higher than in the other area, and the conversion capacity is higher. So this project is telling us how to handle, how to work, which path we have to take, depending on the different areas. And our conclusions, these are not conclusions, but final consideration. 30.16% of the sites is the same or higher than in the case of native vegetation, and this does not exist anywhere else in the world. The root system that we have always discussed is now clear. We do not need to convince anybody else. Third, carbon recovery is not only in the top layers, but up to 80 to 100 centimeters depth. And last but not least, the calcium management and the silicon management. So there is the alternative to increase apart from carbon. And lastly, carbon-based no-till system, what we call the 12 R's, reinvents the system, rethink the actions, reconstructs all processes, recycles what you have to recycle, revitalize the biological cycles, reconnects chemical, physical, biological attributes, reorients the actions for the in the case of the producer, revalues the land, reactivates all relations, and lastly, regenerates. So if it regenerates, it preserves. And if it preserves, we keep the soil and the life. And this is our group. So here on top, the Brazilian team that works with me, and on the lower side of the slide, the Ohio State University, with a special acknowledgement to Professor Lal, because since the first day they believed in this idea, and they put all these researchers into this project. So we now begin the game. Thank you.
Para frente, para trás.
Cambiamos.
Pronto? Buenas tardes. Vamos dar continuidade às charlas. Não existe.
We will continue with the presentations. Good afternoon. No, this is not my presentation.
Você quer passar ela na frente, daí vamos trocar? Não precisa trocar, levar o pendrive. I need to change the pendrive. Vem na frente. Esse, desculpa, aqui.
¿Cuál es cuál?
Adelante.
Perfecto. Bueno, buenas tardes.
Good afternoon. It is a pleasure to be here to discuss the research that we do at INTA and the progress that we are making. And just to tell you more about this, at INTA we have different programs that have to do with the countryside. One of the programs is the Natural Resources Program, where the research is based on several pillars, not just the soil, but also biodiversity and issues related to climate, water, environmental manager, plant protection, elements, and soil remediation, with the objective of maintaining our productive potential, improving it, and recovering different areas. In the research lines of INTA, we have 3 projects at a national level. One has to do with chemical, biological, and physical fertility of soils, the other one for greenhouse gas emissions, and the other one related to carbon sequestration and the capacity to neutralize soil degradation. And in this project, we have 4 main pillars. One is on monitoring soil organic carbon, and the purpose is to increase the presence of carbon in the soil, modeling to estimate potentials and different scenarios on how we could improve these carbon reserves in the soil, and training. Regarding soil organic carbon, last year we developed a joint work with Aapresid and with farmers and with the Secretariat of Agriculture, and we published this map with carbon reserves in the soils of Argentina. This is a map that is based on empirical data in a short period between 2015 and 2022. We took samples from the soil, and not just carbon. We also measured apparent density of the soil in order to calculate the carbon, and being able to relate these carbon stocks with different types of soil in different ecoregions. Thinking about ecoregions, we have the Pampa as one of the regions that accumulates a large part of the soil carbon in our country. Then we have the region of Chaco, the dry Chaco, and the Patagonian Estepa. Since although they have lower presence of carbon, they have higher concentration. So they are important as a reserve for the country. And what happens with soils? If we think about what we're asking of soils, our agricultural systems in general are from soils that derive from pastures with a good root development and green vegetations almost all year round, but with a great contribution of soil carbon. So this is transformed in the agricultural areas of the country. And why? Here we have the national crop maps, the winter crops, the winter map, which is the one on the right, and the summer one at the center. If we look at the colors at that map, we have a high predominance of soybean in the central area. Then we have blue, which stands for corn, in the borders. But looking at the winter map and thinking a little bit of what my colleague from Brazil was just saying, in the winter, we have very little. We could have more, yes. We could have much more. And for winter crops, we have a large surface of the Pampa, and our mollisols show no crops, no living seeds or no living crops. So what are we asking the soil to do? To do what? And how will we care for it? How are we going to do this? In Argentina, we have a really impressive adoption of no-till. We have an impressive potential to recover carbon in the soil. However, that technology of no-till farming, we are not fully applying it, or we're not applying it in a fully correct manner. Why? Because although we have that large surface of no-till farming, the amount of degraded land is very, very high. It's very high for technology adoption and technology availability. We have problems with dust storms. This one is from December 2020 in La Pampa. We have water erosion, and we have a loss of organic matter when we could actually have gains. So there's a gap there. There's a gap where we could be leveraging and increasing our organic matter. This technology and no-till farming needs our support. And what happens when we don't accompany no-till farming? What happens in our soils, which have a higher content of organic matter than the soils that were presented in Brazil, we have a high content of cations, is the type of original organic matter. Because when this is lost, those aggregates are lost. They become more fragile, and we start having a structure that is called laminar structure, which you may have seen, and it is not on the upper part. We will see that granular aspect on the top, which means that we're recovering carbon. But between 2 and 10 centimeters, we will see a laminar layer. And so we sometimes hear people say, oh, no-till farming recovers carbon only in the surface. It actually has the potential to recover carbon more deeply, but we don't give it the opportunity. So what do we need? We need organic matter in that other layer to start breaking that laminar structure that is created because of the moisture dryness cycle. And we are not providing so much organic matter. We can see the horizontal roots that do not penetrate deeply. So what can we do in the meantime while we recover that granular structure typical of mollisols? Because organic matter doesn't find that enough. It is not enough for those for that organic matter. One of the options, this is an experiment that was done in Entre Rios, which was to work with a chicken layer and cast as well. And what we applied on the surface with that laminar structure, if we could break it to allow the roots to penetrate more and increase the process. So the application of plaster and the chicken layer plus the plaster allows us to break that laminar structure. And this allows us to sequester carbon more deeply. So it's not just in the surface, but also a bigger amount of organic matter, more water infiltration for crops, and therefore the crops can go deeper into the soil. Working in Pergamino, we asked ourselves, what would happen if all that surface that we have with summer crops but not winter crops, all of this fallow, this area of fallow, what would happen if we started seeding a cover crop or a bridge crop? We could recover carbon. Yes, we will notice that in the first 10 centimeters of the soil, although maybe 8 or 10 years will go by. We will not see it more deeply because this is a technology that allows us to increase the amount of carbon that goes in, but we are not solving the problem of the laminar structure. We're just postponing it. So with those measures, we can recover carbon, but it will be slow. It will take a lot of time. What happens if a soil is doing better with the rotation? We have a rotation of maize, wheat, and soybean. And what if we intensify more? We include a pasture, a crop, maybe with a cycle of 4 years where we have live-in roots all year long. In this system, we can increase carbon significantly and maybe if we think about what we do when we use the service crop, it might not be enough to break the laminar structure. It is not enough to incorporate more carbon in the deep layers. But the problem with all of this is that we do not give it enough time. We do not get to the point where we can cover the entire year with living roots. So what do we do if we think about this map, this carbon map? We have a high percentage of carbon in our soils, and we can increase it. We can, because we have soils that can recover organic matter, because these are fertile soils that can accommodate the carbon content. But we need, in addition to no-till farming, we need to start incorporating other practices to help that no-till farming. So the opportunity of sequestering carbon, one can see it in a selfish way, maybe, just to think about the fertility that I will achieve, the extra water that I will have to plant, especially if we repeat the drought cycles that we've experienced. Or it can be construed differently, to start thinking about how to generate wealth. If we increase soil carbon, we create wealth. Wealth that is both for our field, but also for everyone. So it is a very tall order when you say, for example, what have we done? I've planted a tree. I've made soil is another thing. I've made soil more fertile. So it's a challenge and it's an opportunity for farmers to use technology, to use research, and to use those results of the research processes to generate wealth. Thank you very much.
Eddie.
Buenas tardes novamente.
Good afternoon once again. I apologize for my language. I'm going to show the presentation. I would like to thank the organization for the opportunity to show these results that are quite preliminary. And I'm going to switch into English.
I have a module in the project with Professor Juca, where we are seeing the indicators for soil health, and they are complementary for what Professor Juca is doing. As the complexity of the soil sampling and the other issues, we analyzed less areas than from Professor Juca. We did 10 sites in Cerrado biome and 10 in Atlantic forest. Just almost, again, in the same way, comparing the native vegetation, the no-tillage system, and we need to standardize also the terms, but I'm calling as an inappropriate soil management. It's anything that the farmer does that's harmful for the soil health or soil biodiversity. And those are the soil health indicators that we measured in this part of the module, are the earthworms, enchytrates that we call potworms or micro earthworms. The activity of the microbiology with carbon of the biomass and glomalin concentration is the soil structure, and also the levels of pesticides linked with ecotoxicological tests that I will present and will tell you a little bit when they present the results. As I told again, there are preliminary results where we are just comparing the sites that we have until now get the processing of the samples from Atlantic forest and Cerrado. And when we look to the earthworms population, looking for number, the abundance of earthworms, when we look to the 3 sites, so we can see that no-tillage system had a higher number compared with the other 2 systems, and native vegetation with the lowest number. And in Cerrado, again, the no-tillage system was quite the same as the native vegetation. Also, apart as earthworm population, we have the biomass. It is an important indicator because it has a relation with the size of the earthworms and how they can do the work in the soil. So the biomass also again in the Atlantic forest was a higher biomass per square meter, and that's because the earthworms are bigger, and I will show you some photos that you can see the species that we were able to identify. And in Cerrado, the highest biomass was in the native vegetation. And then the managed sites like no-tillage system and the other with an appropriate soil management, they had low densities biomass due the size of the earthworms that is predominant by small species. Species richness, that's quite important, that can bring us about a biodiversity. So no-tillage system in southern Atlantic forest had 17 species, a little bit more as the native vegetation. With a predominance of native species, that's a good indicator. That means that our no-tillage system are keeping our natural biodiversity. And when we go to Cerrado, so again, no-tillage, it's almost the same number of species in the three land use, but the no-tillage system has one species more, but the predominance in Cerrado is from exotic species introduced by man colonization and man land use change. Those is just to see the list of the species, and then that you can see the difference among the biomes. So 23 species were identified in Atlantic forest, half a half from native and exotic species. And in Cerrado, we had only 6 species, almost 3 times less, and 5 of them are exotic, and just 1 probably new species that we are working in the identification. Some photos that you can see of the species that we have already found, those are all native species, so are some already known and described, so some small ones, giant worms like Andorrinus duzeni, and Urobenus brasiliensis, that's quite an interesting indicator because it's a surface earthworm. When we manipulate this earthworm, it fragmented. So if it's living in the surface of the land use, it indicates that it's a good environment for it and is able to survive in this environment. To this side. And some other, those are already new species from Atlantic forest, giant new species of earthworms, and some other smaller one, Glossoscolex, that are quite high distributed in all South country, Argentina, Paraguay, Uruguay, and South Brazil. And from Cerrado, we have this already that we are looking for this species. It's an unidentified, and this small one, the Dicogaster, is this with high abundance that we have found and low biomass is an genus, a family of earthworms that came from Africa and is high widespread in farming land use in Brazil. So going on to the other results, we have the potworms. Just you can see how small they are. They are considered mesofauna. The earthworms are macrofauna, visible organism. Potworms are mesofauna. And the potworms, again, the no-tillage system had the highest abundance comparing with the other land use. And when we go to the genus diversity, they are quite challenging to identify. Those in yellow one, we are not sure from if they are exotic or native. The green one are native genus, and the red one are introduced widespread species. And the first one, Henlea, is the first record of this genus in South Hemisphere. So they are characterized from the North Arctic Hemisphere and was the first time found and both in native vegetation and no-tillage system. And the proportion, and for this one we have just from Atlantic forest the processing for now. The number of genus is quite the same, but we can see that the distribution of this genus is a little bit different among the systems. Although no-tillage system have one more species, the distribution is quite similar comparing with the inadequate management. Going to microbiology with the carbon biomass of the microbiology. So we have in both, in both Cerrado and Atlantic forest, the highest activity was in the native vegetation, but no-tillage systems are showing that they are raising it and coming around with what we have in native vegetation. When we go to glomalina is an exudate from the mycorrhiza. This easy extractable. They are decomposed more easily with this other total glomalin. And from this that decompose more easily, we have the highest levels in the no-tillage system, and from them that stays for more time is stable in the environment, native vegetation, but with the no-tillage system reaching almost the same value. Going to the soil structure, so that's we use a method that's called fast soil structure diagnostic. It's called DREST, Diagnóstico Rápido da Estrutura. It's done visually in the soil. 6 is the rate, the maximum rate that we can reach in this evaluation. So that's because the native vegetation has number 6. In Atlantic forest, no-tillage system is almost reaching the native vegetation. In Cerrado is in the middle and a little bit less than the native vegetation reference. Here, just that you can know, we have in the X it's not come down. You can see the depth. So you have up in violet, you have the native vegetation. In green, the no-tillage system, and you can see when it goes to depth, this rate of this structure, it goes down. And when we go to the great land uses, this has the lowest values, and goes in Atlantic forest, they have more structure, compacture. We can see in some photos in the follow slides. And in Cerrado, they have the lowest rates, but they keep in the depths the same values. Just so you can see how this evaluation is performed. So you have in the first column of both the native vegetation. So you can't see any difference among different layers in the soil. That's because it has this 6 value. When we come to the managed no-tillage system, where you have the rule, you have a difference in the soil structure that then this defines the rate that gave to the sites. And when we look to the graded areas, we can see the structure is completely different. And that is how we reach these values that were presented. And then the pesticides level, we are looking for those because the biodiversity is responsible for all the transformation of soil carbon and carbon storage, and also is responsible for the degradation of these principles that we use. In the same way that, depending how we do this management, they are also harmful for soil biodiversity. We analyzed 12 principles for herbicides, insecticides, and fungicides. Those in green, no concentration was identified by the Atlantic forest. The Cerrado is yet being processed. And those in red one, I will just shortly present you to see the levels of the concentration. But just one point, we don't have standardized levels, what we can say, this level of this pesticide is harmful for soil. We have this kind of levels, at least in Brazil, for food and for water, but no standard level yet for soil. So snow tillage system have the highest level, but we don't know if it's harmful. But those are indicators that we need to be aware that something is happening in the management, and we need to try to reduce the use of these pesticides. So imidacloprida is the first one, atrazina. And just to keep also the awareness that those pesticides, we also found them in the native vegetation. For clomazone and for tebuconazole, for clomazone in both native vegetation, tillage has the same concentration, but low. And for this tebuconazole, the degraded area has the highest level compared with the others. And pyraclostrobin, no-tillage system had also the highest level. Again, as I told you, we don't have standardized level to say if it's bad or not, but we need to keep the eyes on it and have as reference. And linked to that, we have this ecotoxicological test with standard species, where we grow these species in the lab, in the soil that we collected in the field. And the measure used is the reproduction. So it's the number of juveniles, how many juveniles we can count on those soils with the application of those pesticides. So just as we don't have a statistic difference, so it shows that although we have some concentrations, the ecotoxicological test of reproduction shows that the juveniles are quite the same, at least for these species in Mata Atlântica and Cerrado for Eisenia foetida. We use two species, Perionyx cavatus, has a little bit difference in the behavior and then reproduction, with less in native vegetation for Atlantic forest and in the degraded sites for the Cerrado. And another organism used is the colembula. It's colembula, I forgot the other name in English, but it's a mesofauna organism, quite important for soil organic matter fragmentation and organic material. And those also, again, they didn't show so much difference among the land uses. The first column is a reference soil used for this ecotoxicological test, okay? And the next steps is to finish all this analysis. We just finished this field work on end of May. So we have many labs. We are working with 7 different labs and universities to do all this analysis. We need to also do this comparison and division that Professor Juca already did with the climatic zones. Probably we can see other patterns along these results. And also to correlate them with all of them, a characteristic and other chemical, other analysis that we are already finishing, and also with some data that Professor Juca, to see how, which of them are the most sensible for those land use system. And of course, soon we will have more precise results. So that was what I had to present to you. We had over than 15 researchers working, and in the field over than 30 people from different parts of South Brazil helping in this work. Thank you so much.
Muchísimas gracias y disculpa, se nos deja.
Thank you very much. We have many questions for you and for these presentations, but unfortunately we are out of time, but very grateful for your presence. and I apologize, but we have to leave the hall. Thank you so much.